In Chapter 4, we covered the diversity of prokaryotes in microbiology, and in Chapter 5, we will be talking about a sampling of the eukaryotes in microbiology. In lab, we'll be focusing entirely on prokaryotic microbes. But in lecture, especially when we get into the chapters dealing specifically with diseases by body system, we will be talking about both prokaryotes and eukaryotes. And we've covered general structure of eukaryotic cells, and now in Chapter 5, we will cover some of the specific examples of medically important eukaryotes. Figure 5.1 shows an electron micrograph and a light micrograph of the parasite that transmits malaria to humans. And that organism is Plasmodium falciparum. So electron micrograph here, this is the microbe, and there are blood cells along with it. This would be a blood sample from a patient. The pink circles are the red blood cells, and the circles that have spots in them are different life stages of the Plasmodium falciparum. This disease is transmitted by mosquitoes. So mosquitoes are probably the most dangerous animal on the planet. A tiger or a rhino or an elephant will probably stomp you pretty badly, but your likelihood of encountering those animals, unless you live exactly where those animals occur naturally, your risk of encountering them is pretty low. However, nearly everywhere in the world, except for places that have permafrost, there are going to be mosquitoes. The best way to combat mosquito-borne diseases is by controlling mosquito populations. And mosquito nets are one of the most inexpensive and effective means of reducing the exposure to the malarial parasite because it's transmitted by mosquitoes. This is a close-up of skin, and it is ringworm. So it looks like a crusty red ring. There are some dried scaly patches in the middle, but ringworm is not a worm. It is a fungus. Both worms and fungi are eukaryotes, but ringworm is a fungus. It's very common in soil and on animal fur, and that is how we would come into contact with it. It was called ringworm because sometimes this ringed structure appearance, the rash on the surface of the skin, looks almost segmented and looks like there's a worm either right on or under the skin, but it is skin irritation caused by the fungus infecting the skin cells. Giardia we saw in one of our earlier lectures. It's Giardia lamblia. It is eukaryotic. It is a protist or a protozoan. Here it is on a gerbil intestine, but it would look just like us, just like this in us if it were on our intestines. And so we have dozens upon dozens, if not hundreds or thousands, of the parasite attached to the intestinal lining. They are ingested by contaminated waters, and so these organisms live in the feces of a lot of different wildlife, and other wildlife or us that ingest that water can end up with a Giardia infection. And here is an underside of one. They've got a little sucker on them that they attach to your intestinal lining and absorb nutrients. Protozoans are typically classified according to how they move. So they may have cilia, which are these short little hair-like projections that sort of wave back and forth to propel the organism through fluid. They can have pseudopods, and P-S-E-U-D-O, pseudo means fake or kind of like, and the suffix pod means foot. So they have fake feet or foot-like projections, and when they can get their cytoplasm, the fluid inside the cell, to push against their cell membrane, it causes these projections that help them to grab onto surfaces and move in a particular direction. And flagellum we saw in both prokaryotes and generalized eukaryotes. And just like in prokaryotes, in eukaryotes the flagellum is used for movement. However, in prokaryotes, that flagellum either spins clockwise or counterclockwise, like a propeller. In eukaryotes, it undulates, it waves, it does not rotate. So, Paramecium is the genus, the first name. There are different species of Paramecium, and they are eukaryotic, so they do have a nucleus. They have these things called contractile vacuoles that are used to contract and push out excess water or waste. They have a cytostome, so S-T-O-M, same word origin as stomach in us. So this is the cell, cyto, the cell, stomach, where food particles enter into the paramecium. In the amoeba, purple dot is still its nucleus. It also has a contractile vacuole with a different shape, same function for pushing fluids out. Contractile vacuoles in Euglena, similar to those in the paramecium, and they have that flagellum. Euglena, what is not shown here is that they have chloroplasts. So they are similar to animal cells in that they are sort of not as rigidly shaped as plant cells, but they are like plant cells in that they do have chloroplasts and are able to photosynthesize. You don't have to memorize the order and the organisms in which grouping of these eukaryotes, but it is just showing that this branch of that phylogenetic tree, this domain, right? The three domains are the bacteria, the archaea, and the eukarya. This is just the eukarya domain. And there are 6 different groupings, are called supergroups. The dotted lines are showing that it's suggested evolutionary relationships. We don't have the entire genetic sequences to be able to confirm that. And we are down here with fungi and choanoflagellates. Our next close group, we have those slime molds that we saw on the phylogenetic tree of all the domains. And again, don't have to memorize these, but those are 6 supergroups within the eukarya. And they have been put into those groups based on their ribosomal RNA sequences. I've added to this chart that these are examples of diseases that are caused by eukaryotes. We are going to be studying many of these in a lot more detail later in the semester. For now, I've indicated that only the underlined ones will be relevant on our first exam. So I've underlined Giardia, because we've seen that a few times. You don't have to know its supergroup or its subgroup, just that it is a eukaryote. Causes giardiasis. Trichomonas cause the sexually transmitted illness, trichomoniasis. That's what STI stands for, is sexually transmitted illness. I've underlined trypanosoma. They cause African sleeping sickness that we'll see on another slide in this lecture. And the plasmodium we saw in the introduction of this chapter, the Plasmodium falciparum that cause malaria. Many other diseases, we will be studying those in the future, but just to start getting our minds around some important eukaryotic microbes. I've underlined 4 on this slide and let's see, 3 on this one. Red algae, there's 2 different groupings of them. Their cell walls are what we extract to use to make the agar that we use for both agar slants and agar plates in lab. Naegleria is an amoeba, and it causes meningoencephalitis. Itis, it's inflammation. Encepha means brain, and meningo means the meninges. So this causes an amoebic infection of the meninges, which cover our central nervous system, and the brain. And I put this out here. Don't use tap water in your neti pot. If you don't know, a neti pot looks sort of like a genie lamp, but it is used to irrigate sinuses. It can be a very effective tool to help flush out pollen and other irritants that are in the nasal passages and the sinuses, but it's very important that the water is sterilized before you add the salt to it to create a saline solution to irrigate the sinuses. Many others down in here, but I have underlined the nematodes because they cause trichinosis, hookworm and pinworm infections that you can get from undercooked meat, primarily pork, and cestoda are tapeworm infections that can be acquired by eating any type of undercooked meat. We saw slime molds in a previous slide. Dictyostelium discoideum can grow on agar. There aren't a lot of eukaryotic things that grow on agar, like protists don't typically grow on agar. Fungi can, and slime molds can. This is the life cycle of that dictyostelium. Put at the top. Don't memorize the life cycle. This is on here because there are 2 different forms. There's the multinucleate, where it has more than 1 nucleus, and there's the unite nucleate, where there's only 1 nucleus. So this slime mold has a crazy life cycle where it can have more than 1 nucleus in some of its life stages. This is another image of a plasmodium. In this on the right side, the sporozoite is one of the life stages of this parasite, and the cartoon is showing you why these are called apicomplexans, because at the apical end, the pointier end, they have this complex, which is what allows them to enter and penetrate into cells. They are eukaryotic because they are protists. So that means they have their nucleus. We can see their rough endoplasmic reticulum, the Golgi mitochondria, and other organelles. This is a ciliate. It has those hair-like projections, the cilia on the outside that help it to move. The cartoon we saw before was of a paramecium. This is Balantidium coli. The trophozoite is just one of the life stages of this organism. And the second name, the species name of coli, we saw in E. coli, the prokaryotic organism that is found in mammalian guts. This is a eukaryotic organism. Its species name is also coli because it is isolated from the gut. And this is the only ciliate, the only protist with cilia that can parasitize humans. So paramecia, we might ingest it with water, but it can't attach to us or cause any harm, and it would just be shed. The Balantidium, however, would be able to parasitize us. The size bar on the bottom is at 10 microns, so this is at least 60, 70 microns across, so it's a very large cell. Paramecium, we're seeing again, there's the cartoon, and here's an actual light micrograph of it, where the features are decidedly less distinct. But it does have that primitive mouth called the oral groove, or the cytostome, and it does have an anal pore to excrete. So they're not really connected like a digestive tract is in animals. They're separate. There's a groove to bring things in, there's a pore for excretion, and it would be done either by diffusion or exocytosis. So I call it interference contrast micrograph. These organisms are fairly transparent, and so a special filter for contrast is used. This organism also has a cytostome, a cell's stomach. This is Stentor roseli. It has little cilia on its ends that rotate and help to filter food into the cytostome. This is a water mold. These are all eukaryotes. Water mold because the fuzzy hyphae, the structures of the mold, trap fluid and make it watery looking, and it has engulfed a dead insect so that it can extract the nutrients that are left in the shell of that insect. Euglena, shown here, is both a cartoon and a light micrograph. It has an eye spot, so it does not have an eye. It is not an organ that is structured like us, but it is a grouping of chemicals that are light sensing. So it's an eye spot, it's not an eye, it's not even an organelle, but it does have the ability to sense light. It does have chloroplasts. It can store the sugars that it makes using photosynthesis. It does have a nucleus and a nucleolus. We also have a nucleolus. That's where our ribosomes are made. It has a contractile vacuole, and it does have that flagellum. When we look at it under a light microscope, we can see that contractile vacuole, and it is decidedly green. Lots and lots of chlorophyll in its chloroplasts. This protist is Trypanosoma brucei that causes African sleeping sickness, or trypanosomiasis. It requires the tsetse fly as part of its life cycle. So the tsetse fly ingests blood from an animal or human, and that would include this protist. It divides by binary fission, splitting in half in the blood and lymph and spinal fluid of humans. And in the midgut of the fly, it also divides. It becomes the infectious stage, and it can just be passed from person to person via the tsetse fly bites. So this disease is found not only in Africa, but primarily in Africa, because that is where this particular fly lives, and without this fly, the protist cannot complete its life cycle. So this is a nematode, or a roundworm. It's also called a pinworm because one end of it is very, very pointy. It's how it can penetrate through cells, and it's very common in soil. Most kids get pinworms at some point, making mud pies, playing in the dirt. They touch their face and their mouth with their hands, and you ingest that worm. The size bar down here is 0.5 millimeter, so that's 1 millimeter. It's about 3 millimeters long, so it's pretty darn tiny. A great variety of flatworms. The phylum is Platyhelminthes. Helminth means worm, and platy sounds like flatty, so these are the flatworms. liver flukes I've pointed out here. I remember them because they look like little livers and they tend to infect the livers of the animals where they are found. And tapeworms, we mentioned before, can be up to 4 to 10 meters long, so that's 12 to 33 feet. The one in this specimen is 4 meters long, so that's really gigantically huge. The liver fluke has a sucker that it attaches to the cells and can absorb nutrients. The cestode or tapeworm, Tinea solium, which is the pork tapeworm, it has suckers all around one end, the head end, that make it attach to the intestines of its host. It also has hooks in the head that really help it to adhere tightly, and the suckers help it to absorb nutrients from its host. Again, we have a life cycle that you don't have to memorize, but it is showing you that this schistosoma infects both humans and snails. A very odd combination of things. The second stage of the life cycle can directly penetrate skin. These are called the myristidia. They have a spike on them. They are microscopic, and they can not just penetrate snail tissue, they can actually penetrate human tissue, and they are found in fresh water. Tapeworms, we would get it by ingesting undercooked meat. They are shed in fecal matter, could get in waterways. Cattle and pigs ingest the vegetation that's been contaminated by those feces, and then the life cycle can continue. The guinea worm, I believe we saw this in a previous slide. It is a worm. The person who is afflicted by this has this sore, and the worm can be attached to a small stick, in this case a matchstick, and coiled onto it to pull it out. Some fungi, many fungi, are multicellular, like molds, and they form hyphae, those furry strings that you see when you get leftovers left too long in the refrigerator, or agar plates left open too long to the air. And so you can have septate hyphae, meaning they have things that separate them, walls, so you have individual cells forming these hyphae, or you can have non-septate hyphae. Each of these little dots is a nucleus. And we can have pseudo-hyphae, fake hyphae. Yeast cells don't really make hyphae, these long strands, but they can bud and create things that look like hyphae. Histoplasma capsulatum. Dimorphic fungus means it has two different shapes depending on its life stage. In the lung, it can be these individual cells, but if it gets into the lymphatic system, it forms the hyphae. And this organism is found in soil where there's bird or bat feces. If it gets dusty and a person is working in that environment where there is the soil contaminated by the histoplasma, causes histoplasmosis. And so when it's inhaled, that high temperature gets the organism to turn to that yeast that multiplies in the lung, causing the histoplasmosis. Spores that are a reproductive cell in these fungi are produced asexually, meaning you don't have to have two different types of cells to come together. They use mitosis to produce an exact genetic copy and create these reproductive structures. So bread mold, if we were to put that under a microscope, Each of these dots is a sporangium. Under a microscope, we'd see the sporangium. This one little dot has probably hundreds of spores in it. And these are different from the prokaryotic endospores. Prokaryotic endospores are made in response to stress, and it's a way to wall off the genetic material until conditions improve. Spores in eukaryotes are just reproductive structures and allow, in this case, the reproductive structures to be airborne and to spread to other surfaces. There's different kinds of spores. You do not need to memorize the types of spores, but they could be in an ascus and make ascospores. They can be in conidia, so this whole thing looks kind of cone-shaped when it's not under an electron microscope, and that's why it's called a conidia. And we can also have the yeast form. The Candida albicans causes yeast infections, vaginal yeast infections, reproductive yeast infections, and thrush, which would be an oral yeast infection. It's an example of ascospores, where in the hyphae the spores are forming in the ascus. And here, the select groups of fungi. Again, we will have lots of different ones we will see in our disease chapters. You don't need to know the grouping or its spore capacity, but the names of them. The Aspergillus niger is a toxic fungus, and I've circled the Amanita phalloides, which is Amanitis, so it's a mushroom that is toxic. So we have conidia, the cone. So the hyphae is the big one, and the microconidia, the little tiny dots. Again, don't have to memorize the types of conidia, but that it is a type of fungus that can cause skin infections. And dinoflagellates, does look a little bit like a dinosaur, doesn't it? Flagellate, it has a flagellum. There's all sorts of shapes and sizes of these. They might have cellulose cell wall. This one has two flagella, and they have perpendicular flagella that cause the spinning motion of this particular dinoflagellate. I love it. Put up at the top. Kelp aren't plants. They're algae. Well, that's something I didn't know till far too long in my biological career because I focused on the truly microscopic. But the kelps are algae. They don't have leaves or stems, which are structures that are required to be in plants. And they don't have cellulose in their cell walls. They don't have roots like plants do. So there's very different types of algae. We've got green algae, the kelp. We've got the red algae that can be multicellular. In this case, the microscopic ones that are unicellular can cause red tide. Different type of green algae. We saw bioluminescence in a previous lecture. These are diatoms. Their cell walls are made out of silica, so they look like little glass boxes. And so if you've ever heard of diatomaceous earth that you can use in potting soil, you can put it around planting beds to keep the slugs away. You can spread it on carpets and leave it there for a while and vacuum it up to get rid of fleas. The reason why it works in those instances is because of the silica. In potting soil, it helps to create aerated pockets to keep the slugs away. They don't want to slime themselves over that because it cuts them. And in our final picture, we have colonial green algae like Volvox. They have cooperative association between cells, and so they look like these giant floating little biospheres. Chlamydomonas are green algae, and they are also single cellular. So they have the nucleus, they have chloroplasts, they have mitochondria, and again, they have an eye spot, this collection of photosensitive pigments. It is unicellular, so these are not cells that are the eye spot. They are collections of pigments that form the eye spot. I have a big X on this because you don't need to memorize the structure of lichen. But lichen is not a single organism. Lichen is a symbiosis of algae and fungus. So the algae are photosynthetic, so they can harvest sunlight and create the carbohydrates that the fungus needs to eat because fungi cannot photosynthesize. And then the fungi provide a structure, the base for the algae to stick to, and then the waste products of the fungi are carbon dioxide and water that the algae need. And the greater the lichen diversity is in an area means that there is better air quality. In Humboldt County, we have an enormous diversity of lichens, so we have great air quality. I am currently in San Antonio, Texas, which is an extremely urban area, so I have yet to see any lichen. But if I do, I'll let you know how many different types I see. So there's 3 different formats for the lichen. We do have crustose, because it is close to its surface, kind of crusty. We have foliose, which looks like foliage, more like leaves. And we have a fruticose. It's kind of fuzzy. Another picture of a fluorescent micrograph of a ciliate. You can see that they have a pretty amazing structure to them. These little horizontal lines would be the cytoskeleton that is lighting up in this particular microbe. The blue is its nucleus. And this very poorly drawn sketch is something I put in here to remind me that now that we've rapidly gone over both prokaryotic and eukaryotic structure and examples, to kind of put a map together for yourself and draw 2 overlapping circles as a Venn diagram. And in one of these circles, you would put characteristics that only eukaryotes have. And in this circle, in this space, features that only prokaryotes have. And where they overlap, all the features you can think of that are shared by both prokaryotic and eukaryotic cells. And there might be some eukaryotic organisms where only some of them have these characteristics, but they are eukaryotic, and some eukaryotic cells have that feature and no prokaryotic cell has it. And over here there could be some features of prokaryotic cells that not every single prokaryotic cell has, but they are unique to only prokaryotic cells. And later in the week or early next week, I will put my summation of what I came up with for this, and you guys can share yours. And usually students come up with even more features than I do, and so we'll see how that works out.